So just to give you a sense of where we are going with things, we are having a webinar in January 15th at noon, and this is going to be on IEPs. And just a little bit of housekeeping. So we use GoToWebinar for our webinar presentations. You should be able to hopefully see the screen and hear the audio. You can join by telephone or on your computer or iPad or phone. And there should be a questions box where you can type in any questions that you have. We'll take the questions at the end. We'll leave about 15 minutes or so for questions. And you can just type those right into the box, and we will get to those at the end. And our speaker today is Dr. Mustafa Sahin. He received his Bachelor's of Science degree from Brown University and his MD and PhD from Yale University School of Medicine. Dr. Sahin completed a pediatrics residency at Children's Hospital of Philadelphia and child neurology residency at Boston Children's Hospital. He did his postdoc research training in developmental neurobiology at Boston Children's Hospital. Dr. Sahin has established and directs the multidisciplinary tuberous sclerosis program at Boston Children's Hospital. He has received numerous awards, including a Spinal Muscular Atrophy Foundation AAN Young Investigator Award, the 2005 Young Investigator Award from the Child Neurology Society, and a 2009 John Merck Scholar Award. In 2013, he established the Translational Neuroscience Center at Boston Children's Hospital, which aims to expand treatment options for nervous system disorders. So we are looking forward to today's presentation. And let's see if I can hand that off. And you are good to go.
Thank you very much. And thanks for the introduction. Good afternoon, everybody. I hope everyone can hear me okay. As was mentioned, I run the translational neuroscience at Boston Children's, and because of the translational nature of our work, I work with a number of pharmaceutical companies. So I want to start with my disclosures. As seen here, we work with a large number of companies that are interested in central nervous system problems, and I'm on the scientific advisory board of some of them. But the slides I'm going to be sharing with you today are, I believe, not biased by these interactions. Let me start by telling you what I am going to cover and what I'm not going to cover. What I'd like to talk about today is repurposing of drugs, which is defined as the process of finding new uses outside the scope of the original medical indication for existing drugs. What I am not going to talk about much is off-label use, taking a drug that's been approved for one indication and using it for another indication. I just want to spend just a couple of minutes on that idea, because you can currently go to your primary care physician or a pediatrician or your psychiatrist, and he or she can prescribe for you a medication for off-label use. They can say to you, hey, I know that this medication is approved for, let's say, arthritis and knee pain, but maybe it's going to work for your chest pain and angina, and they can prescribe that medication for you. That is allowed under this sort of off-label use, and they will be taking a certain risk, obviously, for prescribing, and you'll be taking a certain risk for trying that medication, because that medication has not been shown to be effective or safe for the problem that you're trying to treat. So I have a slide from the FDA website that I'm sharing with you here that basically says what it means to be approved by the FDA and what off-label use means. And if anyone has any further questions about what off-label use means, I would urge you to go to the FDA website and understand what the implications are about using a compound that's maybe approved by FDA for some indication, but not for others. Now, we talk about FDA approval. I wanted to briefly describe what it means to have a compound have a FDA approval. And this again is from the FDA website. So if something is approved by the Food and Drug Administration for an indication, that means that FDA has conducted the evaluation of its risks and benefits. And for each indication, this really differs. The risks of a drug may be evaluated differently for certain indications versus others. Risk of a medication for a disease that might be terminal or with high mortality may be evaluated differently than something that doesn't have high risk in that indication in the first place. So each drug has to sort of fit into its particular risk-benefit ratio or evaluation. There's also the idea that the decision to use the drug is supported by strong scientific evidence. In many cases, this might be preclinical evidence, not necessarily evidence from humans, but maybe even evidence from animal models or cell culture experiments might also be supportive of the use of this drug. And then the FDA approves some drug labeling of the compound for the providers on how to use it, what dose to use and how frequently to use it, etc. So all of these things are included in the approval of a drug by the FDA. So if you are using something off-label, obviously none of these things or some of these things may not be there. So those are the risks that you have to take into account. And in fact, for diseases that many of us worry about, such as autism, as you know, there are not that many approved drugs. So FDA has actually put out a warning, if you will, about potentially dangerous therapies and products for symptoms such as autism spectrum disorder. And this was a relatively recent warning that's on the FDA website that I would urge you to take a look at it. It talks about relatively high-risk therapies that have been suggested by some providers for treating autism spectrum disorder. So with that introduction, I want to turn to repurposing, which is the main topic of this webinar. I mentioned rare diseases in the title. We're going to focus on rare diseases. In the United States, rare diseases are defined as conditions affecting fewer than 200,000 individuals at any given time. And Phelan-McDermid syndrome obviously fits into this category. But if you take all the rare diseases together, it's estimated that 300 million people are affected by rare diseases. And a lot of times they've been also referred to as orphan diseases. There's been a lot of progress in rare diseases recently. In particular, maybe the poster child of success in rare disease treatment has been the approval by the FDA of now 2 drugs for spinal muscular atrophy, a disorder that causes low muscle tone, weakness in young children, and oftentimes results in those children passing away in the first few years of life. This was a really severe disorder that we have been taking care of as child neurologists, and there was no treatment available for this disorder. And as you can see, in December of 2016, the first treatment called Spinraza or nusinersen, which is an antisense oligonucleotide based treatment, got approved for spinal muscular atrophy, and it really changed the whole landscape of how we take care of children with SMA. Soon after this, the viral therapy by putting in the gene for SMN using a viral-mediated delivery system was approved, and a third oral treatment is now being evaluated by the FDA. So there's been huge progress made in this particular rare disease called spinal muscular atrophy, which was one of the more severe neuromuscular disorders that we take care of as child neurologists. The challenge is that SMA is only one of these rare disorders, and we now know that there are 6,000 or more than 6,000 disorders where there's a molecular, in many cases, a genetic basis for, and only 500 of those have a therapy available for them. So there's a really huge unmet need in rare genetic disorders in terms of approved therapies. So with that in mind, we've been focused on repurposing drugs for these rare genetic disorders. And when I say we, many academic centers like ours at Harvard Boston Children's Hospital, but many throughout the world. Academic centers have focused on this. There are also some biotech and drug companies have focused on this as well. Now, why? Why have we focused on repurposing rather than making drugs from scratch? Well, part of the issue is if you're repurposing a drug, the safety and the dosing is worked out for many of these drugs as they went through some amount of research and development. So this allows for faster development times and reduced risks. I'm going to show you, or try to show you in the next couple of slides, what I mean by that. So this is a slide from a paper that came out 15 years ago, but the idea of repurposing drugs was first really highlighted in this paper. And what they show here is that if you go from just identifying a target to treat on the left side of the slide, all the way going to the market, the FDA Approval and market, that can take somewhere between 10 to 17 years. And the chance of success is 10% or less than 10%. In many of the neurological disorders, it might be even less. And the reason for that is that you have to go through each one of these steps, as you can shown here. You have to identify the target, then you have to make a compound that works on that target. Then you have to optimize that compound. You have to have some information about how that compound is delivered and distributed throughout the body and what its availability at the organ or cells that you're interested in treating this. Then only then do you get into clinical trials, and you have to start from phase 1, which is basically safety information for those trials, and then it will take several years from that safety phase to get to the efficacy phase in many of these disorders. So it's a relatively long haul to get this approved. On the optimistic side, somewhere on the order of 10 years. If you are repurposing a drug, however, you can sort of shorten that process significantly, partially because the target identification, at least the compound identification, some of the pharmacokinetic properties of these drugs, etc., may have already been done for a different indication, for a different disease, let's say. And you can use that information to accelerate your studies to treat your disease of interest. And you can even start your human studies at the accelerated stage if there's enough safety information in individuals that are similar to the ones that have been treated in the past. So that can significantly accelerate the process. In this particular slide, it suggests that it can be as short as 3 years, but it can take as long as 12 years. And it also emphasizes that there might be reduced risk and fewer unknowns because you have much of this work done for a different disease previously. So here are the two slides put next to each other, just to highlight where you are saving time. Much of the time savings, as you can see, is in the development stage, in the clinical trial stage, because you have already some of the information from previous use in human subjects before that could be applied to your disease of interest. So I wanted to, in this next slide, share with you again from the same paper, which is 15 years old. It highlights some of the compounds that have been repurposed. So thalidomide is a drug that some of you may have heard of. This was actually developed for sedation and insomnia. It was unfortunately used. It was thought to be very safe for everybody, and was used in pregnant women in the 50s and 60s, and that led to severe birth defects because of this, the way it works on certain growth pathways. And this led to actually significant improvements in our management of the drug approval process. But aside from that, obviously its indication, sedation and nausea, insomnia, did not turn out to be a great use of this drug. However, later it was found that it might be used for certain forms of cancer, in this case multiple myeloma, as well as a severe form of cutaneous manifestations of leprosy. So that drug, which started out from a very different indication and had certain untolerated, obviously untolerable side effects, could potentially be useful in a cancer, multiple myeloma context, and has been FDA approved for this other indication. The second example is from topiramate. Some of you may have heard of this as an anti-seizure medication, and it's still commonly used for seizures. But as you may know, one of its side effects is weight loss in many individuals. As a new indication, as a repurposing in a way, it's now been tested in obesity as an indication, and it's available in a compound form with another drug as a anti-obesity treatment. So you can go from start from a very different original indication and end up with a new indication, as long as the mechanism seems to work for these indications. So this was proposed as a way to accelerate treatments, and has gotten quite a bit of traction. In fact, NIH, the NCATS, has approved or proposed this as a new therapeutic use for existing molecules program. This is from the NCATS website. Feel free to explore it when you have time. But they have a program to improve the complex and time-consuming process of developing new treatments and cures for disease by finding new uses for agents that have already cleared several key steps along the development path. This is from the NCATS website. So this is exactly the repurposing idea, and the idea of taking compounds that have taken several steps along the R&D pathway and have been studied for certain indications and trying them in new indications. What I want to do for the next 10-15 minutes is to turn to a disease that I worked on for the past decade or more, tuberous sclerosis, and give you an example of repurposing in that rare disease. And I think in some ways might have implications for Phelan-McDermid syndrome or other neurodevelopmental disorders. So tuberous sclerosis complex is a genetic disease. It can be inherited, or you can have de novo new mutations that result in the disease. It's due to DNA variants, genetic variants in two genes, TSC1 and TSC2. There are several non-brain manifestations of tuberous sclerosis, but the main brain manifestations are astrocytoma, which is a type of a slow-growing brain tumor; epilepsy, which affects 90+% of the patients, which can be very hard to treat and can be refractory, medically refractory many times; and then autism spectrum disorder and related neuropsychiatric manifestations. So let me show you over the last, let's say, 10 years or so, how the process of repurposing a drug worked for tuberous sclerosis. So what first happened was that the role of the TSC1, TSC2 genes was identified and mapped to a well-studied pathway called the MTOR pathway. So it turns out that cells control their growth by regulating an enzyme called mTOR that stands for mechanistic target of rapamycin, as shown here. And 5 different groups around the world back in 2002 showed that the TSC1 and TSC2 genes regulate this pathway, put a brake on this pathway. And as you can imagine, if you're missing either one of these genes, TSC1 or TSC2, this pathway becomes overactive because the brake is not working, and you make too much protein and the cell grows. The pathway has taken advantage of these, the cells have taken advantage of this pathway in several different ways. Several of these autism-related genes, such as P10, also regulate this pathway, and growth factors regulate this pathway. And then the energy level in the cell, the ATP to AMP conversion, for instance, also regulates this pathway. I won't go into the details of this, but suffice it to say that this is a very central focus of regulation for cells, both nerve cells and cells outside of the nervous system. Then identification of this pathway really changed the landscape of tuberous sclerosis research, because for the first time, we now understood what these two genes that are mutated in TSC do. But at the same time, we also made a connection to a particular enzyme called MTOR, which has its own history coming from a very different perspective. This is a compound that was identified on Easter Island, on the Rapa Nui in the South Pacific. And the history is quite interesting. So this is where this little island is in the South Pacific. And this is what the compound that ended up being identified is called Rapamycin. It has this very interesting structure that allows it to get into different cells, as well as the brain, past the blood-brain barrier to some extent. The history of rapamycin is quite interesting. In the 1960s, a microbiologist collected samples from Easter Island, and they found that they were able to have a suppressive effect on fungus development or fungus growth. Then the scientists at a pharmaceutical company identified that a bacterium in the soil produced this antifungal compound, fungicidal compound. So it was first thought that rapamycin is predominantly working by suppressing growth of fungus. And when the structures was identified, it was called rapamycin. It turned out that work at the National Cancer Institute and other places showed that rapamycin, aside from its effect on fungal growth, actually could also be an immune suppressant and can also not only inhibit the growth of fungus, but in general inhibit the growth of cells and proliferation of cells. And this led to FDA approving rapamycin as an immunosuppressant in patients who need immunosuppression, such as organ transplants or kidney or heart transplants. Often use this medication and have been using it, as you can see, for the past 20 years. So we actually have quite a bit of safety information, as well as potential risks, benefits, pharmacokinetics of this drug from its use in patients with immunosuppression or organ transplants. Another compound that is very similar to rapamycin, it's really different by a very small change called everolimus, has been approved now on coronary artery stents that can be used to block the restenosis, blocking of the coronary arteries. So in patients with symptomatic heart disease, if you put a stent in to open the coronary artery, if that stent has some everolimus, that allows the stent to work for longer. And this is one of the approved uses of the MTOR inhibitors. And then Afinitor, which is a form of MTOR inhibitors made by Novartis, also known as everolimus, is now also approved for use in advanced renal cell carcinoma. So these were the uses of rapamycin and rapamycin-like molecules in the past. But as I showed you earlier, this connection between tuberous sclerosis and MTOR was made in 2002. And that really changed the way we looked at the pathogenesis of tuberous sclerosis. And obviously raised the question, could we use MTOR inhibitors in patients which don't have this brake working well to partially suppress protein synthesis, cell growth, and the manifestations of tuberous sclerosis that we see in the patients. This effort was led in the case of astrocytomas by David Franz, a child neurologist at Cincinnati Children's Hospital. And David Franz published a landmark article in 2006 in Annals of Neurology, where he took just 5 or 6 patients with tuberous sclerosis and these supramandibular giant cell astrocytomas, these slow-growing tumors that are seen here in the patient in panel A. This is this contrast-enhancing lesion that's causing increased ventricular size on this MRI and causing hydrocephalus. The patient was placed on rapamycin for 6 weeks, and as you can see, within the 6 weeks, the tumor is shrunk significantly and the hydrocephalus is improved. The patient had to come off of the medication for about 4 months because of side effects. And unfortunately, the tumor grew back to its original size, and you had the hydrocephalus develop again. The patient was started on a lower dose of the drug and has done well. So this was a really seminal study showing that what we thought would work in terms of blocking mTOR activity downstream of the tuberous sclerosis genes and proteins in patients with tuberous sclerosis could work for some indications in patients with tuberous sclerosis. This led very frequently in 2010. So this first paper was 2006. In 2010, there's a New England Journal paper by Darcy Kruger and David Franz, again from Cincinnati, a single-site study with just 28 patients, showing that everolimus, the other compound very similar to rapamycin, works for suprarenal giant cell astrocytomas. And they measured the size of the tumor, and they showed that in all patients that were treated with the drug, none of them had growth of the tumor, and in many of them there was shrinkage of the tumor size. And this small study, relatively small study of 28 patients, was sufficient to get FDA approval for everolimus. Now, so as you can see, repurposing can work really well, at least in the case of suprarenal giant cell astrocytomas in a rare disease. And even with a small number of patients, you can get FDA approval in a relatively fast fashion. The second indication that was studied for mTOR inhibitors and tuberous sclerosis that I'm going to highlight here is epilepsy. So essentially 90% of the patients or more with tuberous sclerosis go on to develop epilepsy. And it's been shown that the earlier the onset of the seizures, the more likely that intellectual disability will develop. In fact, in children with seizures before the age of 1 year of age, in this old case series back in 1990s, only 8% had average intelligence. I think our point of view now has evolved a little bit, and we are a bit more optimistic. But again, it's important to highlight that regardless of the exact severity, the earlier the seizure onset, the worse the cognitive and behavioral outcome in patients with tuberous sclerosis. And as I mentioned, 90% of the patients with tuberous sclerosis have seizures, and it turns out that if you try to model this disease in the brain in mouse models, almost every mouse model that's been built, some of it's from our lab, many of it are from other collaborators around the world. Each mouse model that's missing TSC in the neurons, in the brain, or in the supporting astrocytes or glial cells in the brain, develop seizures. So these have been extremely useful for repurposing drugs if you have a pathway that you understand. I'll show you just one slide from a mouse study that we did, almost or more than 10 years ago now, and was published with David Kwiatkowski from Brigham and Women's Hospital here. So what David developed was a mouse model that developed seizures, and what you're looking at is the survival of the mouse. These are the mice that are treated with a vehicle or a control compound, and they develop seizures by about 20 days of life, and they all die from seizures by 60 days of life. If you start treating the mice early in the first week of life, they don't show any seizures, and 90% of those mice survive. If you stop the treatment around day 30, then they start having seizures and they start to die. So this was true for 2 different mTOR inhibitors. So this was just proof of principle that in mouse models, in animal models, you could use a compound that's been FDA approved for immunosuppression, that's been FDA approved for renal cell carcinoma and other indications, and use it to suppress seizures in a mouse model. That obviously led to clinical trials. In this case, I'm just showing you the results from a phase 3 trial that led to the approval of everolimus, the mTOR inhibitor, in patients with TSC. This was a very large trial of 345 patients. Again, randomized placebo-controlled trial in this case, unlike the astrocytoma trial. And the primary outcome was the frequency and the severity of these partial onset seizures in patients treated with everolimus versus placebo. And this led to a high-profile publication because it was successful in Lancet in 2016. Again, it took longer than the astrocytomas in this case to do these large trials, but the results are shown here, and I want to share it with you for informing you of some of the possibilities of what you can see in these kind of trials. So here we're looking at response rate, which is greater than 50% reduction from the baseline frequency. So if a patient was having 10 seizures a day, if they went to 4 seizures a day, that is seen as a success marker on this case. And this is the response rate, the percentage of patients that responded in this fashion. So in the placebo group, the patients that were not taking the active drug, actually 15% of them responded by just taking placebo. In the low dose everolimus group, approximately 30% of the patients responded. And in the high dose everolimus group, roughly 40% of the patients responded. And this, as you can see, there's a dose-dependent effect. The more of the drug that you have, the better the response rate, and especially compared to placebo. And these were significant values. This led to FDA approving the drug. It took about 2 years, but it was approved in April of 2018 as a adjunctive treatment for patients with tuberous sclerosis and partial onset seizures. Obviously, as I showed you in the previous slide, only about 40% of the patients respond to this drug, so the 60% don't respond. I had shown you the mouse slide earlier. In that case, essentially 90% of the mice respond. So things are not as positive or optimistic in the human trials, and there might be a number of reasons for that. But there's definitely room to identify new compounds, maybe repurpose other compounds like rapamycin for seizures in tuberous sclerosis. And I wanted to show you how we are going about doing that as just an academic center. We have developed what I would call a small mouse epilepsy monitoring unit. Many of you might be familiar with hospital inpatient epilepsy monitoring units, where we do 24-hour video EEG to try to identify and characterize seizures and seizure types in individuals with tuberous sclerosis or Phelan-McDermid syndrome, etc. Here's our mouse unit, where the mouse is in a cage that is transparent. There's a video, a night vision camera that videotapes the movements of the mice for 24 hours a day. We also have an EEG on the head of the mice that transmits signals to this receiver plate at the bottom. So we can actually do a 24-hour EEG, and we can give different compounds. And we have mouse models that have seizures with some frequency, and we can give a compound and see if that changes the frequency. And this will be the first way we will show, is there any reason to think that this compound may actually work in the patients as well. We may also, as you know, have the capability to develop human nerve cells in a dish now. This is a relatively new development, and we are using this in addition to this epilepsy monitoring unit in the cages. We can also do some monitoring in cell culture. So we can make human neurons with a patient with tuberous sclerosis or Phelan-McDermid syndrome or any other genetic disorder, and we have ways of looking at the activity of those compounds in the dish. This right bottom corner is showing the firing of electrodes that their neurons are plated on a set of electrodes, and each time a group of neurons fires, that electrode detects it and shows it up at this lightning here in the dish. And you can look at the frequency of these oscillations or these electrical waves, and you can compare them in a patient with tuberous sclerosis versus a control, and you can change the firing of these cells. And we've shown, for instance, that MTOR inhibitors that were FDA approved for tuberous sclerosis recently can also reduce the firing of tuberous sclerosis cells in a dish. So that begs the question, are there other compounds that might work like MTOR inhibitors and reduce the firing excitability of these cells in a dish that might potentially be repurposed in clinical trials going forward? So we and many others around the country have similar efforts to repurpose drugs for these rare genetic disorders. For the last couple of minutes, I want to move away from tuberous sclerosis, which I talked to you about for the past several slides, and turn to Phelan-McDermid syndrome. As you know, there are currently some trials going on in Phelan-McDermid syndrome. IGF-1 was previously studied by the group at Mount Sinai, our collaborators, Alex Clausen and his group. and they have a trial now using recombinant human growth hormone. There's another trial ongoing using intranasal oxytocin. Both of these compounds, both the human growth hormone and oxytocin, are approved for different indications by the FDA. So the safety profile, etc., is relatively well studied. And now they are being studied in Phelan-McDermid syndrome, and the pilot studies are ongoing. If you want to know more about these kind of trials, clinicaltrials.gov is the website to go to. And I just copied the two trials that I just mentioned there. And if you click on those in the website, you'll get much more information about the study, where it's ongoing, why is it being done, what it involves to be part of these trials, what sites are involved, etc. But here's a real-time example of repurposing drugs for a rare disease like Phelan-McDermid syndrome. Since rare disease studies take a collaboration among multiple sites, we took this on about 5 years ago, and we developed, as some of you may know, a developmental synaptopathies consortium that is studying 3 different diseases in parallel: tuberous sclerosis, Phelan-McDermid syndrome, and P10 hamartoma syndrome. And we're actually doing a clinical pilot study in P10 hamartoma syndrome using the same drug that we use in tuberous sclerosis. And our goal is to, in some cases, repurpose drugs, and in other cases, identify biomarkers or outcome measures or phenotypes in these large group of patients that we're studying, such that we can accelerate the development of therapies, and such that we have well-characterized patients, we understand the natural history of this disease, that we're ready for clinical trials as they come up. And repurposing of drugs is just one form of these studies. I've tried to highlight the advantages of repurposing drugs rather than making brand new drugs for accelerating therapies in Phelan-McDermid syndrome and other rare genetic disorders. So I'd like to stop here and turn it back to the facilitator and see if there are any questions that I can address.
Great, thank you so much. So we do have a number of questions that have come in, so I'll go through some of those. And folks on the line, if you have additional questions, feel free to submit them. And we'll take as many as we can in the next 18 minutes or so. So first question we have, are repurposed drugs generally less expensive than newly developed drugs?
It depends on where in the patent life the drug is. Some of the drugs that have been recently approved for a certain indication may still have active patent lives, and those drugs, because they're only made by that company, may have a certain price, and it depends on how they were made. May be more expensive than others, etc. Not always, I should say, but in many cases, if a drug has been approved many years ago for a certain indication, this patent life has gone off patent, that might be a way that is cheaper to make, etc. The price will be lower, but not in every case. I guess that's a long answer to a relatively simple question.
Okay. Next question. It seems that patients and caregivers and politicians accuse pharmaceutical and biotech companies for the high pricing of new drugs. I understand that these drugs cost a lot of money for development. Aren't the insurance companies, so payers, to blame as well if they choose not to cover these treatments? Are payers more likely to cover the cost of repurposed drugs?
Yeah, that's a very complicated question, unlike the last one, I think. And I'm not sure I feel qualified to address all aspects of it, but we all agree that I think the cost of drugs and drug development and coverage right now is a major issue for providers, for obviously patients and families affected with disorders who need these drugs. There have been many examples of very high prices in diseases that I take care of, including infantile spasms and some of the neuromuscular disorders that I mentioned earlier. I think in some ways we are all responsible for this because we all contribute to this economic and political system that results in this. I'm not, everybody has probably a part to play. I'm hoping as we develop more of these life-saving drugs that there will be ways to cover the cost and allow more patients to have access to more of these therapies as we go along. This is not just, unfortunately, a problem for the United States, but the whole world, because we see a lot of international patients trying to have access to these drugs that are potentially life-saving for their children, and that brings together a huge number of questions, not only in the U.S., not only in the developed world, but in the developing world and other countries as well. So I'm sort of not addressing that question directly, but I think it's a very big issue we all have to face.
Yeah, it's a complicated question and complicated answer. Okay, so there was a question that came through when we collected some questions ahead of time. So long story short, there's an anecdotal story that some in the PMS community have heard about a drug that was prescribed normally for PKU. A PMS patient was given the drug as part of a trial and had good outcomes from that, but it's not clear if that drug was the sole reason for the improvement. Phelan-McDermid syndrome experts don't see a significant scientific link, and therefore, to this person's knowledge, there was no further research. Even though there was no scientific link, and since the risks seem low, why not have more trials?
I mean, we learn a lot from anecdotal studies. There's sometimes some of this repurposing comes from parents that have tried some off-label indications. One of the things I was trying to highlight is that there has to be good scientific rationale for a drug to be repurposed. What I was trying to show in this presentation is that for the case of repurposing in tuberous sclerosis, there was a lot of data from previous studies in cell culture, in animal models, that would suggest that they should also work in the human trials, and that kind of repurposing, I think, is much more likely to succeed. If there's a scientific rationale to think a compound may be in the pathway, might be helpful. It makes a lot of sense to have more studies on it, but we have to prioritize our research. Unfortunately, we don't have endless amount of resources to study every anecdotal possibility. So I think we have to prioritize by, what is the scientific rationale? Is there any scientific premise based on what we know about the compound and the pathogenesis of Phelan-McDermid syndrome, whether that compound may work in PMS? And then, take those priorities into cell culture and animal experiments as needed.
Great, thank you. Next question. I know this is probably difficult to answer, but with the trials going on today where we're testing the effect of a drug for the first time on our PMS kids, how much time needs to pass before any long-term side effects before we can rule out any long-term side effects from the drugs?
Yeah, that's a very good question. And this is something we all worry about, obviously, as clinicians and clinical researchers developing these drugs. In many cases, the FDA will actually, even if they approve a drug, may ask for a post-approval data collection. There's a drug called vigabatrin that has been FDA approved for infantile spasms. It works remarkably well for infantile spasms. Unfortunately, it has some side effects in terms of retinal toxicity in the eye, in terms of potential visual loss. So FDA approved this drug after much consideration. In fact, the United States was one of the countries where this drug was approved last, after many developed countries had approved this drug. And then they asked us to collect information after approval. And based on that information, it is now recognized that the side effects are actually not as frequent and severe as we originally thought, for instance. So this is just one example that I think continual follow-up is going to be necessary. There's no way to predict if some drug is going to cause a major problem in 20, 30, 40 years from now. Unfortunately, there's no clinical trial that's going to be sufficient to address those concerns. Luckily for repurposing drugs, for instance, in the case of Rapamycin, we had 20+ year data in at least patients with organ transplants that those were relatively well tolerated. So the more that kind of information is available, obviously the safer the drug.
Okay, and our next question, we have some really good ones coming through. 10% of drugs that start from scratch are successful if they make it through 10 to 17 years. What percent of repurposed drugs are successful?
Yeah, I don't have the numbers for that. I think the numbers for repurposed drugs are smaller in general. It probably depends on the indication and the mechanism of action of the drugs. Unfortunately, I don't have that data.
Okay. And is anything being developed to, quote, turn up the volume of the second Shank3 gene or Shank2 to compensate for the loss of Shank3?
Right. So that's a good question. I'm not going to answer that question, directly for Shank2 and Shank3 for the following reason, because this idea that we have 2 copies of most genes, and in many cases, if you lose 1 copy, you get symptoms. Is it possible to turn on the other copy, or in individuals with X-linked diseases, turn on the inactive X to turn on the expression of some of these genes is a very good idea. In fact, there are some methods that people are developing to do that. And I suspect there's going to be reason to think that some of these genetic methodologies or even small molecule methodologies might be working in the future. I should say the one success story that I highlighted in the very beginning, the spinal muscular atrophy, the way the first drug that got approved for SMA is by turning on the expression of a gene that's right next to it. Not the expression itself, but the full-length expression of the gene, such that you increase the expression. I think that those kind of methodologies will be applicable to Shank3. I think will at least be tested in cell culture, in stem cell models, etc. And there's reason to think that they might be effective in at least some of the childhood neurological diseases, if not many of them.
Our next question. When drugs are initially screened to see if they are potentially effective for a condition and fail, are those failures reported so other scientists won't need to test them, or is the interested scientific community aware through informal contacts?
That's a great question. One of the reasons that the clinicaltrials.gov was mentioned earlier is really a requirement now. If you put your clinical trial on clinicaltrials.gov, that you're required now to put the results of it up as well as soon as the study is done. So this is one of the compliance aspects that we have to follow. So FDA and NIH and other funders have realized that many studies don't get reported, unfortunately, as your questioner asks. And hopefully this will become more transparent, and there's going to be more sharing of data. In the past, I would say many of the failed trials failed, meaning either they did not recruit enough patients to finish the trial, or the drug was not effective, or was not safe, was not really that clear. And then with these more stringent requirements for sharing data and sharing results, hopefully we'll have more transparency.
And our next question, what should be done about the fact that there are no consistent biomarkers for PMS that apply to rodents, especially since different mutation models don't have the same cellular phenotype?
Yeah, that's a great question. I think many of us in the field are trying to develop translational biomarkers for PMS and other childhood neurological disorders. I would say we are in the relatively early stages. I wouldn't necessarily give up. We're trying to develop better models of showing Shank3 deficiency in the brain in different developmental stages. I don't think we've looked enough at the use of electrophysiological biomarkers or other ways of imaging the brain that might be useful. I think there's a lot of actually activity in this field right now, and I'm quite optimistic that we will hopefully either using the iPSC-derived neurons in the dish or in the animal models, predominantly mouse so far, but there are also rats and other animal models being developed, that we will find some translational biomarkers. I think we need those biomarkers to be able to have confidence in the treatment trials that the compound is engaging the target. at least. And as you know, the Achilles heel of our field is the outcome measures. And without the biomarkers, our confidence in the outcome measures is not likely to be complete. So the combination of biomarkers and outcome measures hopefully will lead to positive trials.
Any chance of research from different countries like Israel, Europe, and America working together to shorten the amount of time this all takes?
Well, especially for clinical trials, we probably have to do that. There are many examples I was giving for the epilepsy trial in tuberous sclerosis was a international, multi-continent study, and they got 345 patients in a relatively short period of time to be able to complete that study. So I have no doubt that we're going to have to work with our collaborators in Europe, in Middle East, and other places. There are some, at least for investigator-initiated trials, there are some new regulations in Europe that partially make this difficult, but I have confidence we can overcome them.
Do patients with tuberous sclerosis that do not have early seizures ever go on anti-seizure meds as prophylactic treatment? Would you recommend this for PMS patients?
Great question. Yes, in tuberous sclerosis, actually, I did not get a chance to talk about this, but we formed a consortium to study this a few years ago. There were similar effects identified in Europe by Sir Józwiak and his colleagues in Poland. And what we did was to get an NIH study that was before this developmental synaptopathies consortium that I mentioned. And first thing we did was to look for a biomarker for impending seizures in TSC patients. And we identified a signature, a fingerprint on the EEG that tells you that the patient is about to have seizures in the next couple of months before the actual clinical seizures start. That's not always possible for the neurodevelopmental disorders, but in TSC we are lucky because many of the patients get identified at birth because they have heart tumors. So we actually did a study where we did EEG prospectively on patients born with heart tumors and tuberous sclerosis, and then found this signature on the EEG I mentioned. So as a result of that, we are doing a prevention trial using an anti-seizure medication in patients that are asymptomatic. They don't have seizures at this point, they just have abnormal EEG. We're testing whether using this anti-seizure medication as soon as the EEG becomes abnormal before clinical seizures start is going to result in a better outcome. We don't know the answer for that yet. The study is just about to finish in terms of enrollment this year, and hopefully we'll have results in the next couple of years. I don't think we have the same information for Phelan-McDermid syndrome right now. It's something that needs to be tested. Unfortunately, not all of these anti-seizure medications are without side effects or other medications that one might imagine using asymptomatically. So I will be very careful about doing that unless we have good reason, good either preclinical or clinical data, hopefully in the near future, to test these kind of questions.
When should groups like Phelan-McDermid Syndrome Foundation get involved in drug pricing and access? How would the foundation best advocate?
Oh, I think, the example I mentioned to you about SMA, there were more than one foundation or advocacy organization that helped with the process of getting those drugs developed as well as approved, and they have been quite active in working with pharmaceutical companies. I'm not sure what their advocacy, how effective their advocacy has been in terms of pricing. That's a much more complicated question, but in terms of drug development, I think the patient advocacy organization and foundations have a huge role to play, and they can. It can be something as simple as obviously help enrolling patients for clinical trials, getting the word out about clinical trials, getting the community interested, educated, be willing to part in placebo-controlled trials. These are all huge steps that in some disease communities, unfortunately, have not been developed. I think the Phelan-McDermid Syndrome Foundation has done an excellent job so far in terms of getting the community aware of the clinical studies that are ongoing, getting the community ready for future clinical trials, what it means to be a partner and a participant in a clinical trial, what the consent process involves, what it means to be on the placebo arm. And these are really important educational aspects that that PMSF is doing a great job in.
And then kind of as a follow-up, what can the average parent do to help and to advocate?
Well, I guess people are already calling into this webinar, which is a good start. I think being aware of what the options are out there, being aware of some of the off-label use issues that are happening. We have advocated, for instance, in our clinic and our community in tuberous sclerosis to make sure that parents are aware of the research advances, but be also mindful of the fact that some of these advances have not been tested in patients yet, and not ask for off-label treatment from their physicians until the appropriate clinical trials are done, to take part in the clinical trials, especially the ones that are placebo-controlled, where we can actually compare the effects to those that are, that can be due to just the natural history of the disease, and educating each other, educating the wider community, sometimes educating their own physicians that they take care of them about the advances in this field, and make them aware of the clinical trials. I think those are important steps you can take part in as a single family.
Well, we are at the hour. I just want to thank our speaker so much for taking the time for this presentation. I want to thank our families and our community for joining the webinar and for all the great questions. If you have any additional questions, feel free to send them my way, and we can try and get those answered. I know there were a couple we did not get to. My email is Jackie, JACKIE, @pmsf.org, and we can try and get answers for you. There will also be a post-webinar evaluation that goes out once you leave the webinar. So if you could take a couple minutes to fill that out, that would be very helpful for us. And we also record all of these webinars. So if you want to listen again, if you want to send it to a friend, family member, give us a couple days and we will have that on YouTube on the Foundation's YouTube channel. And we look forward to having you for future webinars. Dr. Sahin, thank you so much for your time today.
Thank you very much for calling in. Thanks. Bye-bye.